Hello, I'm Brianna Zarati, and my research proposal will be focusing on regional variation in gonad length and weight of the channeled whelk species. Some background information before we dive into our research question and hypothesis. So, channeled whelk are large predatory marine snails typically found in the eastern coast of North America. Females are typically larger in size than males. For reproduction purposes, females carry fertilized eggs internally. Once these eggs are laid, they're laid out in long capsules. So the picture on the right hand side actually depicts what that looks like. That long slinging thing are capsules of eggs. There can be hundreds of eggs laid at one time. However, only a few eggs per capsule actually hatch. Some factors affecting female gonad size include having warm, stable temperatures. So this ensures that there's faster gametogenesis, which usually results in larger gonads. Having abundant food availability is also important. These species can have more energy, which also could lead to larger gonads. And then environmental stress. So the less stress that these species endure due to their habitation and the environmental factors around them could also result in larger gonad size. So for the purpose of this project, I really wanted to look at how does region affect this species' gonad length and mass. So specifically, when we talk about regions, we're going to be focusing on three different regions offshore of Massachusetts. This includes Buzzards Bay, the Vineyard Sound, and Nantucket Sound, and those are depicted on the top right-hand picture there. You can see that they're pretty close together, but they actually do have some differences in climate and temperature. So for the hypothesis, I believe that female channel whelk inhabiting the Vineyard or Nantucket Sound will have larger and longer gonads because these regions offer more stable temperatures, more abundant food availability, and low environmental stress compared to Buzzards Bay, for example. So diving a little bit into the data and methodology. So all of the data is going to be coming from the mother database or the multi-species ovary tissue histology electronic repository from ASU. In there are 7 mature female donor samples of this species, the channeled whelk species. Each sample contains some metadata on the XML files that include the region of collection, the gonad weight, and the gonad length. Additionally, they have some information about other variables that will be necessary to consider any confounding variables such as things like body mass. So for the methodology, I really plan on extracting the data from the samples, the metadata. So this could look like using some of the Unix methodology that we've used in this class previously. From there, in R, running a Kruskal-Wallis test, as well as generating a gonadosomatic index. So this is going to be the gonad mass divided by the body mass per sample. So this will give us, since it's such a small sample size, this will give us a great idea of exactly how the gonad lengths and sizes are being affected by region, not only by region but also by body mass. Like I said earlier, this is going to be one of those confounding variables. And lastly, here are my references. So the first reference there has a lot of information about the mother database. The second has a lot of information, some of that background information that we talked about earlier about the channeled whelk species in general. And then the last one has some information about the factors that affect gonad size in females.